Good afternoon. This is Dr. Dan Guerra, and you are watching Authentic Biochemistry podcast. Today is, I guess, the 27th of August, 2023. So we're going to be continuing on this biomedical portrait. This is number 2, and I believe it's chapter 4. Remember, the overall topic is sexual dimorphism and bioenergetics. So I thought we would do a video because I think I have the studio pretty much set up now, and we'll see if this all works out in terms of quality when we're finished with it. So I think we can, with all of that aside, you know that I'm Dan Guerra, and you know that you are about ready to get this lecture. So hopefully that's what you came for. All right, so we did that. All right, so yeah, here we go. First of all, when we talk about bioenergetics, we have to discuss primarily at the beginning, what is the carbon source that's going to be oxidized to generate metabolic energy in the form of ATP to do work, chemical work in the body. And we can think about stored carbon sources, such as adipose, which is primarily triacylglycerol. Or we can think about storage glycogen, which is a homopolymer of glucose. So when we do bioenergetics lectures in biochemistry, we tend to have to go into metabolism before we actually get into what are the specific quantifications necessary to be able to do particular functions physiologically in the body. So that's what we're going to do today. We're going to go from discussing general aspects of male versus female biochemistry physiology, and then inserting that into the background of general intermediary metabolism and then associated bioenergetics. Okay. So the first slide basically is giving you that very simple understanding that glucose can be converted to lactic acid. That would be one of the end products of anaerobic glucose oxidation, and that would be called glycolysis. Lactate can also be used as a substrate to synthesize glucose, and that process is called gluconeogenesis. Now, glucose can be stored, as I said, as a homopolymer with alpha-1,4, alpha-1,6 linkages of glucose, and that would be called glycogenesis. And then glycogenolysis would be the breakdown of storage glycogen that you could find in skeletal muscle of the liver, and then that would regenerate glucose. Okay, so it just gives you a very, very small sketch of an understanding of how glucose is utilized and the various pathways we might need to bring into view to be able to examine them. So let's talk about some basics here. And again, I do a lot of writing on these slides so that you can have them as a video, and I'm going to read them to you. Okay, sorry. Otherwise, I would just have my mug up there and I'd be reading to you anyway, so you might as well see the words. So what have I said here? Glucose metabolism in both sexes is highly responsive to various physiological and nutritional states and, of course, physical fitness. So there is a precondition of how glucose metabolism functions, such as muscle mass, fat mass, age, and, as we will find out, male versus female. And that can be the basal understanding, in fact, basal energetics. But we also have to think about what are the physiological responses that are in queue, that means in line, right? When we're examining it. So, for example, what about a viral challenge? What about a bacterial infection? What about a cut? What about exhaustion from running a marathon or climbing up a mountain or taking a 10-mile walk, right? So those would be responses to physiological activities. We also have to think about nutritional states. So that means overall, what's the BMI? The BMI, of course, is the height to width ratio. But we also have to think about what is the basal metabolic rate, the BMR, and that differs in age distribution, in weight, that means in mass, and also in height, and furthermore in the state of the skeletal organs and the cardiac muscle. Okay? So there's a lot of things right with that first basic we would have to unpack. So during exercise, women, and this is in general, will oxidize more lipids and less carbohydrate. Now that's during exercise. That's not at the basal energetic state. Okay? And because of oxidizing more lipids than carbohydrates, they will deplete less muscle glycogen and liver glycogen, and they will exhibit a reduced basal hepatic gluconeogenesis. So less glucose will be synthesized in the liver because there's more beta oxidation going on. As an aside, in high altitude environments, like above, say, 8,000, 9,000 feet, women adjust to their carbon source utilization more efficiently than men do. So this has a lot to do, you remember, with 2,3-bis-phosphoglyceric acid and overall hemoglobin production to be able to transport molecular oxygen where oxygen tensions are decreasing above, say, 10,000 feet in elevation. So that is all playing a role. That would be one of those physiological responses that's also linked to environment. Right? Now, the preferential substrate selection is attributed to estrogens versus androgens. We've talked already a little bit about that in our previous biomedical portrait on postmenopausal Alzheimer's disease. Remember the estrogen receptors, alpha, beta, the GPC estrogen receptor versus testosterone and overall androgen reception in the male. We were only talking about the female in that first biomedical portrait because we were talking about postmenopausal women and Alzheimer's disease, right? But we did cover some of this back then. And the first couple of lectures on this second portrait, we've also gotten involved in it. So, sex-specific physiological sequelae to, for example, exercise, that could be diet there too, or aging, right? Or any other aspect I just brought up, will include a lower sympathetic neural activity and a greater type 1 and type 2 muscle fiber density in women. Okay, so that's one of the sexual differences. Now, human bioenergetics is differentially attuned to male versus female, absolute sex-specific functions that would support what? In utero, post-parturitional, pubescent, and adult developmental biology, which would involve, of course, unique requirements for females in gestation, lactation, skeletal muscle mass production and maintenance, lipid deposition, and what we had talked about in that first portrait, CNS specific requirements. Okay. Another basic is that male metabolism is in some ways considered simpler. It can be more variable. Remember, males tend to have more variation, and that is linked to that heterogametic theory, right? XY versus XX in females. But nevertheless, it does not involve a monthly turnover, which would, of course, be the menstrual cycle, females, and thus has been argued in the literature to manifest a reinforced default state. By reinforced, I mean a more control in such a way that the changes between, say, glucose oxidation versus fatty acid oxidation move slower. at least in terms of skeletal muscle. Now, when you're talking about lipid versus carbohydrate metabolism and the lymphatic and leukocyte populations in the body, the immune cells, if you go back and listen to my immunology lectures, either the immunoepigenetics we just finished, those 100 lectures, you'll know that there is a great deal of specificity of carbon utilization, whether or not you're talking about T cells, B cells versus plasma cells, which are, of course, antibody producing, and then all of the innate immune cells, right? Whether or not, for example, macrophages are M1 or M2, and that would include, of course, microglia and the CNS, et cetera, et cetera, et cetera. So all of those things we have to take into account when we're describing this biomedical portrait. And we're not going to be able to do all of the detail on all those parameters. I'm synthesizing it in way of summary now. Okay? But we need to understand that that's what the general components of the literature suggest, a reinforced default state in male. Now, these differences are the consequence of the action of sex chromosomes and associated sex-specific hormones, including the estrogens and progesterone in the female and the androgens in the males. And of course, there are complex receptor transcription factor mediated responses. And of course, the expression of those proteins, right? All right. Now, in humans, sex-specific specialization is associated with distinct adipose distribution within the body. And the preferential utilization of glucose, amino acids, or fatty acids, those three carbon sources, for ATP production. So, the sensu strictu of bioenergetics. Right? Females store more lipids and they have a higher whole body insulin sensitivity than males. Again, these are generalizations, but they're generalizations that are penetrant. Okay, we see them whenever we look for them. Okay, so keep that in mind. More lipids, more total body lipid, and also more insulin sensitivity, okay, than males. And males tend to oxidize more lipids, nevertheless, than females, particularly when we're talking about BEE. Remember the energy expenditures we talked about recently? So BEE is basal energy expenditure. I wrote it out for you. Okay. Men possess a lower percentage of fat mass, even if you're looking at male versus female obese populations of similar body mass. Okay. They still have lower percentage of adipose than females when both are obese or both are lean or both are in between. Okay. Now that is a data point that again is solvent and penetrant. That means it has a mainstay in the literature. Okay. Now, with that, we also find there's more type 2 diabetes with common pathobiochemical insulin resistance in men. Now, why? That almost seems like a contrarian in a way, because type 2 diabetes is really a disease of lipid metabolism. But it's the part of metabolism we have to think about, not simply lipid mass, you see. And that's, of course, the details are always more important than the generalization, right? As we shall see. Now, women generally have a more robust, as we've been saying, basal insulin sensitivity. And there's been some arguments in the literature about that. We'll go through some detail with it as we move through these lectures. But it might be due to the fact that there is this menstrual cycle and this overall bioenergetic differential, which must occur because of that cycling, at least into menopause. And that involves glucose versus lipid for bioenergetic needs, with amino acid metabolism playing a role typically in stress responses, and also when there is a sparing of protein, sparing of lean body mass, until the point where nutrition doesn't fulfill that requirement, and then protein breakdown starts to occur. And this can occur in some forms of stage 3, stage 4 cancer, the cachexia, wasting stages of cancers. Okay, so those are some basics that I think are important. Now, take a look at this diagram here. Forget where I got this from, some textbook, right? So let's look at how different cells metabolize carbon sources. Red blood cells primarily use glucose, and they convert it to lactate. But red blood cells also, so that would be a glycolysis that would be running in RBCs. But red blood cells also run the oxidative pentophosphate shunt. Okay, now, why do they run the oxidative pentophosphate shunt? One of the products of that pathway is, of course, ribose biphosphate, which is a unique pentose, right? And you know what ribose biphosphate would be used for? DNA and RNA synthesis. Now, that was a red herring because red blood cells lose, remember, their nuclei when they mature to reticulocytes. They're just transporting oxygen. So there's a massive amount of hemoglobin. But they still need a carbon source, right? They still have to have bioenergetics. And so what am I saying here? I'm saying that when oxidative pentophosphate shunt is working, why is it working? Well, because the oxidative pentophosphate shunt produces NADPH, and NADPH will be the ultimate reductant of reactive oxygen. And you can generate a lot of reactive oxygen, which is nothing more than not completely reduced forms of molecular oxygen, right? Incompletely reduced forms of molecular oxygen are reactive oxygen species. They are radicals, right? They have unpaired electrons, right? So you need a lot of NADPH to quench all that ROS in the red blood cells. And that's why active OPP is there. Now, what about in the brain? What goes on? Well, we know the brain uses glucose as its primary carbon source. Right away, you know why, because I've said it 100 times, but I've said it once over the years. Because lipids don't generally pass the blood-brain barrier. Generally. Now, that's a key word there. We talked about senescence. We talked about the apolipoprotein E4, remember, coming from what lipoprotein fraction? HDL, right? That's right. And you know that lipoproteins are the traffickers of lipids. Why? Because lipids are insoluble in aqueous. And serum is primarily an aqueous solution. Right? It is, yes. So lipids need to be translocated with proteins. So lipoproteins find it difficult to move through the BBB because that's a very impermeant membrane, not only because of the size and the aggregation, the hydrophobicity, et cetera, of lipoproteins, but because anything of that mass and probably that diameter could also be bacterial cells, right? Or virions, or fungal spores, or fungal masses, right? Or any number of aggregate proteinopathies. And certainly none of that would want to be normally allowed through the BBB and into the fine workings of the neural network, right? So glucose primary carbon source, although remember we talked about ketone bodies. Remember acetoacetate and beta hydroxybutyrate, right? Those are generated from fatty acid oxidation. They can come from skeletal muscle or liver primarily, or kidney. But what about those ketone bodies? Remember, we also talked about the fact that you can synthesize those ketone bodies from the degradation of sphingomyelin from myelinated axons using the astrocytes. And that ketone body utilization in the brain, it also occurs with simple long-term fasting and starvation because glycogen reserves get depleted. And if you're starving, that means there's no nutritional input of glucose, correct? So what you're going to be using then is depot fat stores, adipose tissue. And so the more depot fat you have, all things considered, probably the longer you would be able to last without any nutritional input, not considering things like essentials in the diet, essential amino acids, essential vitamins and minerals, essential fatty acids for that matter, right? Right. And water balance and whatnot, mineral balance. Okay. But you know there's ketogenesis in the brain because we covered it. But the brain basically is just completely oxidizing whatever the carbon source is going to be, whether it is occasionally amino acids after transamination. Most likely it's going to be glucose. So you do glycolytic pathway, and rather than going to lactate, you'll go to pyruvate, pyruvate dehydrogenase, pyruvate dehydrogenase to acetyl-CoA. Also, you're going to be running some of that pyruvate, some of it, to kickstart the TCA cycle with the pyruvate carboxylase. So you make some acetic acid in the mitochondria, in the neuron, and then what it'll do, you run the TCA cycle, you make a lot of NADH, NADH2. Those will be reoxidized in the electron transport chain, all in that intermitochondrial membrane. And what you're going to do with the proton pumping ATP is make the ATP necessary to run all that neural activity while you're learning biochemistry. Right? And what else would you be doing, right? All right, now what else is going on? What's going on in the adipose? Well, in the adipose, you've got glucose being converted to glycerol, right? Remember, that's the alcohol. And glycerol plus fatty acids can be used to synthesize storage depot fat, the neutral lipid is called, triacylglycerol. Remember that adipocytes don't so much divide, although they can divide, particularly in their pre-terminally differentiated state. Remember that? Yes. But when adipocytes are fully differentiated, they primarily just grow by accretion of triacylglycerol. Right? More and more lipid body formation within the adipocyte. Right? Okay. Now, what's going on with the liver is far more complex because the liver is a importing, exporting organ, always. The liver isn't so much taking care of what's going on for the liver, like the growth of the liver, or some kind of specific function, right? Like what the lungs do in terms of taking in molecular oxygen and respiring carbon dioxide, among a lot of other things the lungs do, right? Yeah. No, the liver primarily is a metabolic organ. So look what happens. So you got down here, you're looking at intake into the liver. You've got glycogen breakdown. Glycogen be broken down in the liver to glucose. We already talked about that. Glycogen phosphorylase enzyme. Glucose then can be transported out of the liver because the liver is one of the few places, really the only organ in the human body, which has a glucose 6-phosphatase. So remember the end product, maybe you don't remember because we haven't gone over it recently, but those of you that are really good budding biochemists do remember that the end product would be glucose 6-phosphate, glucose 1-phosphate and glucose 6-phosphate. So you need a phosphatase to have the glucose leave the liver. So see, that's how glucose is taken out of the liver. It's got to be dephosphorylated. If it's phosphorylated, the glucose is trapped there. Again, we can talk about lactic acid, some lactic acid being formed in the RBCs. If lactic acid is completely soluble in circulation, it can make it to the liver, and that is one aspect of gluconeogenesis. It's gluconeogenesis there, right? Of course. Proteins can be broken down. What kind of proteins? Well, any proteins that are endocytosed, such as low-density lipoprotein, such as apolipoproteins coming from receiving HDL or VLDL or IDL or VLDL, IDL, or LDL or HDL back to the liver with lipid cargo mostly removed except for the HDL fraction. Those apolipoproteins have to turn over. Remember, many of them are synthesized either in the small intestine or the liver, right? Those apolipoproteins. They have to turn over. Why? Because proteins auto-oxidize when in circulation. Why is that? Because there's a lot of molecular oxygen in the serum. Okay. So those amino acids then, those proteins can be broken down as amino acids, can be transaminated, and transamination, like alanine, can go directly to pyruvate, so you're already at pyruvic acid. But you remember we talked about amino acid utilization being either gluconeogenic or ketogenic. Remember that? Way back in biochemistry lectures, not that long ago for me, because it seems I'm always doing those intermediary lectures. because I'm always doing biochemistry lectures. But anyway, the liver's doing that, taking amino acids, converting them to alpha keto acids from the TCA cycle, or for alanine, go directly into pyruvate. And then, of course, the pyruvate converted to acetyl-CoA. And then what else is going on in the liver with excess carbon source? You're going to be synthesizing triacylglycerol in the liver as well. So that's one of the ways you get a fatty liver. Are there sex differences between any of these things I've been talking about? Yes, there are. Even though a liver is pretty much a liver, and a red blood cell is pretty much a red blood cell, male versus female. The reason there are differences is because there are subtle distinctions in the transcriptomes, proteomes, and metabolomes of all these organs in male versus female. Why is that? Developmental biology. Remember, from conception to right now, when you're ancient, like way past my age, obviously, but when you become ancient, you still are terminally differentiated, right? And so all those organs that you've been carrying with you all these years, your liver, your kidneys, your lungs, your CNS, right? Your pancreas, your small intestine, all the organs and all the cells in the body, yes, they turn over. Some cells much less than others, of course, to prevent what? Cell proliferation, leading to what? Precancerous state, right? So, but that means that all that terminal differentiation has occurred. Remember, terminal differentiation is set up into a developmental sequeon based after conception on whether or not you're male or female. So there's not just those sex organ differentiation, but when the sex organs are differentiated, they become part of the overall metabolic grid. And of course, the endocrine, exocrine, and autocrine systems for the body, and also the immune responses, right? So there are female versus male immune system specificities. That's why there are certain types of immune-linked diseases that are higher in men versus women. Whenever you think about why is cancer higher in a particular cancer, of course, prostate cancer would always be higher in men, and breast cancer, even though men do have sort of the breast, not developed the same way, of course, because again, of sex hormones and sex chromosomes for that. But nevertheless, those organs are differentiated differently, terminal differentiated differently. And when the cells are replenished, they're replenished according to that sequence of events. Right? So what I'm giving you here, I'm also now throwing in part of that BioMedical 2 lecture, right? But what I'm showing you here is just what I would show you in general biochemistry if we were starting off, say, the spring semester and doing intermediary metabolism. I'd show you a slide just like this, right? Actually, one with more detail about pathways and whatnot, but we're not doing that here right now because these are just vignettes, right? These are short lectures, what I'm calling portraits because I like the term, right? All right, what else going on there? So we're talking about glycogen. Some glycogen can be stored in the liver. When excess lipid is stored, you can get fatty liver. When does that occur? When there's an excessive amount of carbon entering in the form of lipid or ready to be synthesized into lipids, say from glucose, or even from glycogen to glucose to fatty acid to triacylglycerol. And that occurs in dyslipidemia. Where do you get dyslipidemia? Obesity. Aging associated or not. Senescence associated or not. Right? You understand? So obesity is one of those chronic lifelong diseases if a person isn't able to keep their body mass down. They can't keep their body mass index down, which isn't the greatest parameter I know, not the greatest metric I know, because people who build a lot of muscle have pretty high BMIs and they're certainly not obese, right? So it really has to do with lean body mass versus fat body mass, right? That's a better parameter to really talk about. But even that has some equivocation, and I brought it up and I will do it in the future. All right, what else am I going with? Muscle. Well, the muscle is an organ that has to do physical work, right? So there's got to be a lot of proteolysis, breaking down to amino acids. Those amino acids like glutamine and alanine, right? They can enter the bloodstream. And when they enter the bloodstream, they can go to the liver, and they can be used as a carbon source for gluconeogenesis. Because remember, the muscle does not carry out gluconeogenesis. Right? The muscle is using some storage glycogen for sure. So a person is really built up in their pectoral muscles, what you're seeing there is a lot of buildup of protein mass, but underlying that also is a lot of oil lipid bodies and glycogen, storage glycogen. Right. So when you're utilizing glycogen, when you're utilizing your skeletal muscle mass, for example, when you're chopping wood or cutting down trees, right? My favorite activities, particularly not chopping down, but cutting down trees, right? Chopping down with an axis. It can be done, and I've done it, but it's quite labor extensive, I would say, right? And plus, anything bigger than wrist width trees, you don't want to be chopping those trees down. You want to be cutting down the chainsaw. Anyway, doing a lot of physical activity, you're going to be taking care of your glycogen metabolism real quickly, and you're going to need some aerobic metabolism because you need molecular oxygen for beta oxidation of fatty acids. And that's going to be the whole lipolysis of triacylglycerol, utilization of fatty acids, and you're going to be generating some ketone bodies out of that muscle tissue, right? While you're also generating lactic acid, core recycle lactic acid back to the liver. What's lactic acid being used for in the liver? Gluconeogenesis. Okay. So the muscle is doing that. So the muscle is producing a lot of carbon dioxide as well. And of course, water is the other byproduct of complete respiratory pathways. So kind of like the brain, right? But the muscle stores triacylglycerol, and from intramyocellular triacylglycerol. Okay, and that's in the form of perilipin-coated oil droplets or lipid bodies, right? Remember that. Keep on emphasizing that because it's amazing how many people don't know that. A healthy muscle has a lot of lipid, but not lipid that's, how shall I put this? Intermyocellular. So intramyocellular lipid is good because it could be, if everything is sufficiently oxidative, you have plenty of molecular oxygen as well, you can carry out beta oxidation. Excessive muscle activity makes the muscle anaerobic. That's why you can't just use lipid all the time, because lipid would be the preferred energy source. Why? Because once the lipid's stored there, you have to worry about translocation, which is slow in circulation, obviously. But the reason it would be preferred is because you get a lot more ATP per mole of fatty acid than you do from glucose, remember? But that's not the case because the skeletal muscle becomes anaerobic with a great deal of contraction cycles. And because of that, when oxygen becomes limiting, right, the blood vessels then contract, right? Less oxygen transport. You have to rely then on amino acids. So there's some muscle protein breakdown, and we know about that. The myosin protein is broken down and resynthesized, right? From amino acids, transfer RNA, the whole bit, and a translational process. So why I showed you all of this is give you the idea. And this is not, again, I say this a lot in lecture. This is a sketch of what's going on, but it is an authentic sketch, something that we've studied for 100 years. We have a really good idea what's going on. There is tissue specificity for carbon source for bioenergetics. That's what all this says. But there is communication throughout. Of course, the body is what? It has to be homeostatic. The body has to integrate bioenergetics in the liver, in the kidney, in the brain, in the heart muscle, in the skeletal muscle, in the small intestine, in the large intestine, in the lungs, all the time, 24/7, all the way from conception to death. Okay? So there's always communication. There's always interaction. And those interactions, those event ontologies, are once again, because of the variability in men is higher than women. That's where you see distinct differences in the microutilization of carbon sources, for example, and also the macroutilization, not just for bioenergetics, but for, say, the immune response. Immune response for, say, dealing with an infection or dealing with a precancerous growth or dealing with cardiac hypertrophy or dealing with neurodegeneration, see? And those differences then are mapped out according to things that are associated with pathophysiology, like being overweight or being too thin or being malnourished, something that used to be much more common than it is now, but still occurs, of course, in a great deal of the world. But malnourished also means obese. You're just overnourished, right? Still malnourishment. Okay. So that's mostly what causes disease, relative, of course, to reactive oxygen, single nucleotide polymorphisms, changes in gene expression because of epigenetic phenomena that can either be normal physiological or pathophysiological in nature. And then, of course, when cell cycle starts to get corrupted and you don't go through cell cycle, so you get a lot of what? Apoptosis, or maybe necrotosis or ferroptosis, which can lead to hyperinflammatory responses, which can lead to disease states, high morbidity and mortality that way too. Right? So you can see how all this is integrated. That's why there would be differences in pathophysiological states in all of these systems I'm talking about, this brief sketch I'm doing. But then because of the developmental process, starting from conception between male and female, all the variations are also going to be built in those two axes. And because of that, we can talk about differences in nutrition, talk about differences in which diseases affect men over women. And again, not just organs that are specific to a male versus a female population. Right? And we're well aware of this, right? Of course we are. There's also a lot of that standing error in what should be, what one would consider to be a normalization of longevity. The fact that men tend to die before women. Well, part of it has to do with bioenergetics, and not storing as much lipid. But then when a man becomes obese, his obesity can be more profoundly morbid than the female, because the female has a better control over lipid deposits because female carries more total adipose throughout her life. See how that works? And that has to do with insulin. It also has to do with hormone-sensitive lipases and the adipokines coming from the adipose tissue, and also from other adipokines coming, for example, from the stomach, like ghrelin, and all the regulation of metabolism at the level of the endocrine level, hormonal level, but also all the inflammatory systems coming from the lymphocytes and leukocytes. Right? So I know I took a long time with this slide, but I wanted to make sure that if we're going to do a video, I want to emphasize by specifically telling you that these lectures we're doing are only going to be a portrait, right? One beautiful, painting by Raphael or something, right? Or by Da Vinci. And that's it. We're not going to be able to do 1,000 lectures, but you could easily do 1,000 lectures, not 100. Just like immunoepigenetics, we only stopped at 100. We could have stopped at 10,000. And where do you get these numbers? Where do I get these numbers? The published scientific literature. Constantly having new contributions every day. And then you might say, well, okay, we keep up with the new contributions and we have different subdisciplines. Yes, that's true. But what are we doing in authentic biochemistry? We're trying to tear away those walls of disciplines. Yeah, I'm just a biochemist. I'm not a real physiologist. Real physiologists are real physiologists, but they're not biochemists, you see. But biochemists, because we're down there at the foundation of living systems, because everything in the cell is biochemical by nature, and everything is physiological is biochemical, and everything is genetic is biochemical, right? What do you think DNA and RNA metabolism are? It's all biochemistry. Right? Even when we're talking about, DNA replication, DNA recombination, DNA repair, or the transcription of RNA, mRNA, tRNA, ribosomal RNA, microRNAs, right? Long non-coding RNAs. It's all biochemistry. So biochemists have to take into account all of that. And so we have to read a larger amount of literature to cross disciplines, huge disciplines in biomedicine. For example, the oncologist versus the cardiologist. I can switch from cardiac muscle to a brain tumor in 30 seconds or less, right? Because we have to be able to talk about what is the consequence of a brain tumor to the cardiac muscle. And we have to make it coherent and authentic relative to what we know about all these pathways. And to keep accounting of that, you have to read tremendous amount of literature. So again, what I'm saying, the reason that textbooks don't cut it, reason that simply getting synopses or abstracts of papers don't cut it. Remember how we go down into detail about how papers are written in authentic biochemistry, right? I don't do it enough, but I do it enough, more so than others. And what am I talking about? What are the techniques that are used? When is mass spec used? When is gas chromatography versus liquid chromatography? What's the utility of polyacrylamide gel electrophoresis, or using a Western blot versus a dot blot, or using cytohistochemistry instead, right? Well, you need to know what all those different lab techniques are. And you don't only need to understand how those techniques are carried out, you need to know when they're appropriate or not. And when you're reading the paper, you need to know if they were done appropriately, performed appropriately, according to domain and function of whatever's being studied. All right? So you have all this literature out there, but it cannot be synthesized into a one-page report every day, even keeping up with the constant introduction of new papers every day. And then there's all of the papers that have already been published. Now, again, you might consider that all those papers are just there and we can go back and we can access them as scientists. And yes, that's what we do. But before there were computers, I said this many times, so I'm old enough to remember. It's like it seems like it was yesterday when I had to get a paper. If I didn't have a subscription to the journal, and I couldn't afford to have subscriptions to any more than one or two, my colleagues might have subscriptions to other journals that I didn't have, and we'd share them in that one like that, the hard copy of it. But we'd have to go to the library. And not only go to the library, we would have to photocopy papers. How many papers do you think are going to be in the library photocopying? Well, you could spend all morning there doing that when you were writing for a lecture. Yeah. That's right. That's how come professors get so old and gray so quickly. We were always doing that. But nowadays, you have it on the computer. Does that make it easier? Well, I don't have to go to the library and use a photocopy machine. But all of the papers being there don't mean anything unless I read them. And how do I know which ones to read and which ones that I don't necessarily have to read? For example, for today's lecture. Well, I have to be fine-tuned in my knowledge base, don't I? And I have to know when to examine something in detail and bring it out, especially when I'm teaching it. Because if I'm teaching it, I'm only giving you the synthesis and not the analysis. I'm only giving you the final summary and not all of the dialectical interpretation in between. Then you're taking it on account of my abilities to be able to be presenting what is authentically there as a subsection of what is published. That you then know something about that given thing, such as sexual dimorphism and metabolic disease. But I can be flawed. Of course I'm flawed. So what I try to do is make sure that I know it at the level that I would need to know it to teach it. Right? Not just for myself, to be able to explain it to others. That's why classrooms are much better than doing this recording like this, or even if it's online and we're doing it live. And I've done a lot of that over the years too. But in the classroom, it's much more spontaneous because someone could stop me right now and ask questions, right? And that's a better way to learn. It's more of a Socratic way of learning. But we do what we can, right? So the reason I'm telling you all this, and I know I've said it before, is you have to take a lot of this for granted, that medical doctors are constantly reading, and maybe in some of them, not many, are actually contributing to peer-reviewed literature. Some do, but most don't. Most that deal with patients don't. And so it's not that they, should, because they don't have the time to do that, because what they are medical practitioners, right? They're dealing with patients. They're dealing with writing prescriptions and diagnosing diseases. So whose responsibility is it to keep up with the literature? Well, it's them to some degree, but nothing like what a scientist does, what a research scientist in particular. Not somebody that calls himself a scientist who's an administrator, or that just, has a suit on every day and they administer science from some office. No, a scientist that actually knows how to work at the lab bench and knows how to read the papers. And what new techniques came out since I haven't been at the lab bench, which has been a while, not that long really, then I have to understand those new techniques. And most of the new techniques are really just modifications of older ones. Just like when I was in the laboratory for 30 or more years, that's what we were doing too. We would read older papers, modify the technique, and then sometimes put it in our publication as a new technique, but we would, of course, reference it, right? So, but if you know your chemistry and you know your biochemistry and you know how to work in a laboratory, you know how to make buffers, and you know what pH is, ionic strength is, and what temperature does to reactions, and how to prepare membranes and do all those sorts of things, not only can you perform that in the laboratory, you can also interpret whether or not it's done coherently in the papers you're reading. Right? And again, I'm saying all this in video form because I don't emphasize it enough, I think, even though I bring it up probably more often than some care to hear. So what I'm saying is, we're going to go through this portrait. Let me see what the next slide is. Might stop here anyway, but let's see what the next slide is. I'm going to go through this portrait, but I'm only going to spend 10, 12, 13, 15 lectures on it. Now, no more. But I want to use it then as a chapter, if you will, in a book, and the book is Biomedical Portraits, 2023. I don't know, July till December 2023, whatever you could call it that. And it would only be for that time. But you can go back to that and you can say, okay, here's a chapter Guerra was doing, on sex dimorphism and metabolic disease in 2023. Now it's 2025. These topics, now we can run those topics in a search engine, right? And look for new papers published in it. Or when I give you the references, use those references as jumping off points to look into that literature from that corpus of literature that's already been published. All right, so that's what we're doing here. I think I won't do this muscle adenylate ratio because it's going to take me at least 5 minutes to explain just this one slide. And I really think I should stop at this point because I want to make sure this video lecture came out okay. So again, the reason I'm doing video rather than audio just right now is because it's been suggested to me from colleagues who I respect that I might reach a wider audience if I do more video. Because most people, and people have told me, they like video versus just listening to audio podcasts. And I fully understand that, right? I would prefer if we were just in a big lecture on, we could meet every day. Fine with me, but where would that be, right? So we're going to stop here. And next time, I cannot promise it will be another video lecture. It might be, okay? We might be able to pull that off. I don't know. Meaning I might decide to do that, okay? Do another video lecture back to back. But today's Sunday, and the week I tend to have other duties as well as I'm writing. So I have writing duties, which I'm pretty regimented, trying to get more regimented on. And also I have the audio lectures. The reason the audio lectures are easier for me, not easier to prepare for, but easier to deliver, is because I don't need all this production to get the video to work. It does take time. And sometimes if the quality isn't good, then the performance doesn't look good, then people aren't going to learn what I'm trying to teach. That's then defeats the purpose, right? All right. So I'm going to stop here. We only got to a few slides into this lecture. But this is going to be a lecture I'm going to be feeding off of for a lot of the audio ones from the podcast. So thank you very much for listening to the podcast. I know pretty much what countries check in on authentic biochemistry. I get that kind of information. I don't know much else other than that, how many total people. But I can tell you once again that this is a labor of love. I love doing these lectures. I really am very pleased that the podcast is doing so well in terms of quality and acceptance. I guess it was number one on some recent survey of a biochemistry podcast. So thank you very much for that. Let's keep it up. And I will try to do more video. But all of this does take time. And time means that I have to pull myself away from what I'm otherwise doing. We all have many things to do. And that means I'm not getting compensated when I do this. So any way that you can add to the piggy bank or to the tin can. Imagine me on the street corner giving you these biochemistry lectures. How much coinage you would throw in the tin can, for example, for today's lecture. Right? And you can do that via PayPal. And you can also do it directly on the podcast. There's a place for you to subscribe and to put money down there. I really appreciate it if you can help me with financing this, because I would like to grow it, not because I need a bigger audience. I don't care really. I'm happy with the audience I have, but that we can continue to do this and do it more often, not just do it almost 4 or 5 times a week, but absolutely 5 or 6 times a week, because I would be more than willing to do that, because again, it's a labor of love. And I'm humbled by the fact that you listen, and I'm also humbled by what we're doing here, which is discussing the beauty of nature. Dr. Dan Guerra from Authentic Biochemistry Podcast Studios in northern Idaho, saying bye for now.